Hi everybody, welcome back to another video. In today's video we're going to talk in quite a bit of detail about the phospholipid bilayer, or the plasma membrane of the cell. We'll talk about the structure of it and the function of it. More so, we'll talk about how it allows for selective permeability, aka its functions in membrane transport, and we'll also touch on the fact that it helps anchor cells together through cell-to-cell adhesion. Okay, if you guys learned something from the video, if it truly helps you understand the topic better, please consider liking, consider subscribing, and maybe leaving a comment. And without further ado, let's dive right into it. So just to get you guys oriented to this diagram here, okay? Here is going to be a portion of the cell membrane, all right, with a plasma membrane. This portion over here is the intracellular space, okay? So this would be your cytosol, and over here would be the extracellular space, okay? So you guys know that the extracellular space, or the extracellular matrix, is made up of many things, okay? So it can be made up of a meshwork of fibrous proteins, namely collagen, okay, maybe some fibronectin and elastin, okay. But there's other things as well, and elastin, okay. And we know that all of this stuff is kind of just floating around in the fluid that surrounds the extracellular matrix, which is called the interstitial fluid, okay. Alrighty, now the collagen is really just to provide kind of a tensile strength. All right, it's actually the most abundant protein in the body. The fibronectin that surrounds, again, the outside of the cell, right? This is more so promoting cell adhesion. It basically just holds the cell in place. And actually, reduced levels of this have been linked to metastasis of cancer, so basically the spreading of a tumor. And elastin, which kind of gives the ability of stretch and recoiling. All right, you'll find this a lot in the lung tissue, okay? It's very elastic. Okay, now that is the kind of structure of the extracellular fluid, and we already know what the intracellular fluid is. It's pretty much just this watery gel-like substance called the cytosol, okay? And by the way, the interstitial fluid it's not just these things. There are other things that make up it, okay? But for the purposes of this video, we'll just stick to that, okay? And just know that it is a kind of a watery gel-like kind of fluid, the interstitial fluid, and it is definitely composed of all of these kind of proteins, which, if you're curious, are secreted by a specific cell type called a fibroblast. Okay, great. Fibroblast makes these things, proteins. Alrighty, now let's go ahead and start talking about all of this stuff that's kind of embedded in this membrane and what functions do they have. Well, first of all, we have to look at the constituents of the membrane, right? And they seem to be these little things over here with like a little ball for a head and these little tails, okay? So let's kind of zoom in on one of them. And if we zoom in on one of them, here's what we're going to see. You guys will remember from the cell bio video that they have a polar hydrophilic head, right? Hydrophilic means water-loving, okay? That is negatively charged, okay? And sometimes, I said, this head can contain molecules such as choline within it, okay? And then they have these negatively charged tails, non-polar tails that are uncharged, okay? Sorry about that. Negatively tails, non-polar tails that are uncharged, not negatively charged. I don't know why I keep saying that. And these guys are definitely hydrophobic. They hate water, okay? That's very important, okay? So the head is hydrophilic, the tail is hydrophobic. Alrighty. Now, what are these tails actually made up of? Well, they're made up of one thing, actually, and that thing is going to be called a fatty acid, okay? So they're made up of fatty acids, but we have two types of fatty acids. We can have a saturated fatty acid, and we can have an unsaturated fatty acid, okay? I'm just gonna write that, okay? Now, the difference between these two is really in the way they are formed. Unsaturated fatty acids, they kind of have like a kink to them, which forms like a U-shaped structure. Saturated fatty acids are more of like a linear chain molecule, so boom, 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 boom. Something like this would be a saturated fatty acid, whereas a unsaturated fatty acid would be going like this, okay? And then there'd be like a, like a U-shaped structure almost, okay? There's a kink, there's a bend in there. And that's due to the presence of a double bond, a covalent bond in there. Now, how does this affect the actual phospholipid? Because you guys remember this whole thing was called a phospholipid. Okay, how does that affect it? Well, remember that we said in the previous videos that the membrane is just a whole bunch of these phospholipids that kind of self-assemble into a lipid bilayer, okay? They literally always self-assemble into this lipid bilayer shape, okay, where you have the hydrophobic tails pointing inwards and the hydrophilic heads pointing outwards, respectively, to the extracellular space and the intracellular space, okay? Well, if there was a lot of unsaturated fatty acids that make up these tails over here, okay, what would end up happening is that the membrane would end up being very, very much fluid, okay? So a lot of these guys would increase the membrane's fluidity, okay, meaning that there's just a lot more kind of space for molecules to move through, and the membrane just kind of moves around way more, okay? Because the lipid bilayer, it's not just like a block of butter, right? People think of it as like, okay, it's made up of fat, so it has to be kind of like the solid structure. It's really not. It has more of like an oil consistency, if anything, because it's super like wavy and flowy, and all of these kind of proteins and whatnot that are just studded in it are like icebergs in a sea, okay, where they can just kind of move around in this ever-changing and flowing phospholipid sea. Okay, so the presence of more unsaturated fatty acids within these phospholipids can cause the membrane to have higher fluidity, and vice versa, more saturated fat would actually kind of prevent this, okay? And again, this just has to do with their structure. Unsaturated fatty acids, they're more further apart from one another, so the membrane becomes more fluid, whereas saturated fats are kind of like more tightly packed with one another, okay? All right, so that's the effect of these saturated and unsaturated fats on the fluidity, okay? And this kind of puts a thought in your head of diets, right? So if your diet is full of high unsaturated fat, you might have a more fluid phospholipid bilayer, okay? All right, what other things can we find inside of this membrane other than the main structural component, which is these phospholipids, okay? And, I've already talked about the fact that these phospholipids are intrinsically going to have some sort of selectivity already due to their hydrophobic nature inside and hydrophilic nature outside. So I'm not going to retouch on that. If you're curious, go watch the previous videos in this playlist. Okay. Let's take a look at the next thing that is kind of in this membrane. The next thing that we can kind of see is this molecule over here, and this, guys, is cholesterol. Okay, so both this guy and that guy, they're both cholesterol. And cholesterol, again, it's kind of intercalated in between these little phospholipids here, and its presence also contributes to membrane fluidity, okay? Whereas if you have high amounts of it, it can actually lower the fluidity of the membrane, and this kind of makes sense. It makes it more rigid because, again, it's kind of wedged in between these phospholipids, and so if there's a lot of it, you can imagine that they're kind of just stopping this kind of flowing movement by getting in the way. However, if there's actually lower amounts of it, this cholesterol, you actually increase the membrane's fluidity. Okay, so that's just a note about cholesterol. Now, the other thing that you'll notice is these phospholipids that just kind of are all around, they're not just stationary, These guys, they can not only twirl, okay, so they're spinning constantly, okay, they can vibrate, they can switch places with other phospholipids. In fact, you can actually tag one phospholipid and see it traveling all throughout the membrane of one cell. okay? And sometimes, in very rare instances, they can actually even flip to the other side, okay? But that requires specific enzymes that are actually called flipases, okay? I'm not even kidding. Now, that's that note about these phospholipids again. The other thing we can see is this kind of Y-shaped structure here, okay? What the hell is this structure? All right, this guy right here is known as a receptor. Now, some of you might not know what a receptor is, okay? But a receptor is essentially something that picks up a signal and then carries a specific function that that signal tells it to. What is the signal that it's picking up? Well, this receptor is picking up a signal in the shape of this chemical right here, this triangular chemical right here. It's called, the triangular chemical, the umbrella term for it is called a ligand. So a ligand will interact with its receptor, and then the receptor will pick up the signal from that ligand and turn it into a function. All right? And notice that there's this kind of lock and key mechanism where the ligand has to fit the exact shape of this receptor in order to activate or turn on this receptor. Okay? Now, just to give you a kind of interesting and a real world example for this, okay? I'm sure everybody has heard of the hormone insulin, okay? Insulin is responsible for what? It's responsible for lowering blood sugar. You know how it does that? That hormone insulin is considered a ligand. So let's say that hormone insulin will be our ligand, and it will bind to its receptor on the cell membrane of a specific cell, okay? And once it does that, it will activate that receptor, and that receptor will then in turn tell the cell that, hey, you know what, you guys need to put up specific protein channels on this membrane to suck the glucose out from the blood. Okay? And what happens then? The blood glucose levels will then drop back down to normal. That's how insulin works in a very basic sense. It is a ligand that binds to its receptor, and the receptor, after being activated, will carry out the function that that ligand wanted to do. Okay? And you can see how the membrane plays a very big role in this by actually containing these receptors. Okay? Great. So that's the other thing that we want to talk about. The other stuff I want to talk about is that the membrane has many proteins on it. In fact, it's made up of a whole bunch of proteins, a whole bunch of lipids, and a whole bunch of carbohydrates, okay? And while there are about 1 billion phospholipids per cell, and the plasma membrane has about 50 times more lipid molecules than protein molecules, the actual weight of the bilayer is really determined by the proteins because they're just so much larger than the lipids, okay? And here's one example of a protein that you might see inside of the membrane where we call it an integral protein, okay? Integral protein. This just means that this protein is crossing all throughout the membrane, okay? So it doesn't cut off halfway. It actually goes through the entirety of the membrane, all right? And this protein can be anything. It could be, I don't know, an enzyme, okay? But then there's other proteins that are not integral, and they are actually called peripheral proteins. Okay, and again, these peripheral proteins, they don't go all the way through. They're actually attached to only one half of the membrane, and they don't go all the way through. And a great example of this can actually be found inside the cell in a membrane-bound organelle called the mitochondria. If you guys remember, complex 2 of the mitochondria contains a protein complex that does not penetrate the entirety of the membrane. It only goes halfway through. That's why it can't pump protons into the intermembrane space. Okay, so that's another example, okay? And peripheral proteins can be other things, right? Sometimes they can be just membrane-bound enzymes, okay? All right, what are some other things that we can talk about? This other one is pretty interesting, and we'll talk about it more in the later halves of the video. In fact, we'll talk about most of these more in the rest of this video. This one is a interesting one. It's called a docking marker acceptor. Okay, now what does the word docking mean? You guys ever heard of a ship that's kind of pulling into the dock? Okay, what is it doing? It's kind of stopping and anchoring itself there. Okay, Now, this is a docking marker acceptor. The best example I can give you is this. Let's say the cell has some cargo, okay, in the form of this vesicle. Now, this vesicle, on the outsides of it, will be told it needs to go into a very specific location, okay? And that is by the presence of these signals, okay? Or more appropriately, docking markers, okay? Now, this vesicle, let's say, contains, I don't know, a receptor that I want to put on the cell membrane. It has a docking marker outside on this vesicle, and this docking marker will come and fuse or bind to this docking marker acceptor. And once this happens, this membraned vesicle will fuse with the cell membrane, and guess what happens? The receptor will now kind of join the rest of this lipid bilayer, okay? Great. And again, we'll talk about this in more depth later in the video, but that's what docking marker acceptors are there for. Alrighty. Next, we're going to have, let's change the color here. We're going to have ion channels, okay? Or channels in general, but I'm just choosing to call it ion channels, and there's a reason for this, okay? So, ion channels specifically, or channels in general, they're actually very narrow and very small, okay? They're about less than or equal to 0.8 nanometers, okay? That should be nanometers, all right? And they really mostly allow for small water-soluble molecules, mainly like ions to come in, okay? So again, things like sodium that are normally in a high concentration outside of the cell will come in through this actual water-filled pore or a channel. This is very much so different than this other molecule called a carrier protein, okay? Carrier protein. This carrier protein quite literally is supposed to transport a very specific substance. So let's take our favorite molecule here that we use for energy called glucose, okay? This glucose will have to come and bind into this pocket, okay? The circular pocket here, right? And as soon as it binds here, this carrier protein will change its shape. So guess what? This area of it will now close, and this one will open to the inside of the cell. And then that glucose molecule can now go in, okay? This is very much different than the ion channels. One, because the carrier protein is actually bigger, okay? And two, the carrier protein is actually changing its shape to facilitate the movement of this molecule inside, okay? The other thing is that carrier molecules exhibit specificity, okay? And not only that, they exhibit a couple of important characteristics. They exhibit specificity, like I mentioned. They exhibit saturation, okay? And they exhibit competition, okay? So let's kind of demonstrate this in a very simple example, okay? Let's say I have a cell, okay? And this cell has one carrier on its surface, okay? And this carrier is, you know what, look, they're here, right here. This green molecule is going to be glucose, and this red molecule is going to be fructose. Let's say this channel right here, or sorry, the carrier, is only specific to glucose. And so watch this. This glucose molecule, it'll go inside of the cell. But what about this fructose molecule? Can it use this same carrier protein to enter the cell? No, because this molecule, this carrier protein, is only specific for glucose. So that is one variable checked off of our list. Specificity. These carrier proteins are specific to specific molecules. Maybe there's a fructose carrier. Maybe there's a glucose carrier, okay? Maybe there's an amino acid carrier, a fatty acid carrier, okay? And these things do exist. Let's do another example, okay? Let's talk about saturation. Okay, this one is actually a very simple concept that very often gets confused. Let's say I have I have the cell membrane, right? This cell, here's a nucleus, and this is the cell membrane, and this is a carrier, this is another carrier, okay? Let's say I have 1 and 2 glucose molecules, okay? And this one is currently choosing to go through this carrier, and this one is currently choosing to go through this carrier. Notice how when I have 2 molecules of glucose, I can easily transport them in. But what if, instead of 2 molecules of glucose, I had 1, 2, 3, and 4? What now? Would I be able to transport all of my glucose molecules in through these guys? No. And why will I not be able to? Because currently, at this current moment in time, my transporters are busy carrying these 2 glucose molecules. Which means what? Which means my carriers are saturated. They are busy, okay? So that's what saturated means, okay? It means the carriers are currently working at a maximum rate. They're frankly very busy. And what ends up happening to these glucose molecules? Will they get transported? No. And so, sometimes in physiological examples, we might be talking about the kidney, where from the actual renal tubule, we might want to reabsorb some glucose back into the blood, but if those glucose transporters are saturated, we might actually not be able to reabsorb the glucose and end up, excreting it in the urine. Okay, so this is a very real example that can happen. Okay. And one last example that we can use is the example of competition. Okay, let's get rid of all this. And let's say I have this transporter. This transporter, right, it can bind both glucose and it can bind both glucose and fructose. So this guy is fructose, which is another sugar, and this is glucose. Okay, this carrier, it has the ability to bind both of these guys or carry both of these guys, but one at a time. Which one is going to win this competition? Well, there's a couple of ways that this can go. If the carrier, for example, has a higher liking or a higher affinity towards glucose, then it might tend to favor glucose transport more than fructose. Or if it has equal affinity for both of them, maybe it's a numbers game. Maybe there just happens to be more fructose molecules, and thus they tend to go in more than the glucose molecules, okay? This is this concept of competition, all right, where these 2 molecules are competing for the same transporter, and different factors can determine which one actually wins in using the transporter or carrier. Okay, so now you understand that these carrier proteins, they exhibit very specific properties, and we can actually demonstrate one of these properties using a graph. So let's say on the y-axis, we have a graph that talks about the rate of transport, okay? So the rate of transport of a molecule. On the x-axis, we have the concentration of that molecule. So low concentration, high concentration. So if I go from left to right, the concentration of that molecule on the x-axis is increasing. So let's say this is 0, this is 10 of some arbitrary unit, okay? And again, this is a 0, and maybe this is like 30 of the rate of transport of some arbitrary unit. Okay, great. And sorry, this would both be 0 here. All right, now let's see what this graph actually is talking about. So this graph is saying that, let's take a molecule. Let's talk about glucose. Okay, I just picked a random molecule. This graph is saying that if I were to look at 2 different types of transport, one being the one in green called simple diffusion, okay, and the other one in this purple being facilitated diffusion, we would witness 2 very different things. Okay, so let's take a look at simple diffusion. And by the way, what does simple diffusion actually mean? Simple diffusion means that something can move in and out of the membrane without any help by any, for example, protein or anything else. So let's take the example of a well-known thing called oxygen. You guys know oxygen? It doesn't have a charge, and it's very lipid soluble, so it can just easily move through the membrane in and of its own accord. And the only thing it needs to move through is a concentration gradient. you guys already know that from my previous video on metabolism that things require a concentration gradient to move through a membrane. And we'll talk about this more in later in the video when we talk about Fick's law. But things like to go from a high concentration to a low concentration. Okay, passively, no energy required to just move down their concentration gradient. Okay, so oxygen, okay, loves to go down its concentration gradient into the cell. Now let's take a look at this. Okay, in a low concentration scenario, right, you can see that the rate of transport is pretty low. But as the concentration of a molecule increases on one side of the membrane, okay, so for example, the outside of the membrane, the rate of transport of that molecule goes up in simple diffusion. Okay, so notice, over here to here, over here to here, over here to here. It's directly proportional with the green line. This means that as long as my concentration of substance, okay, goes up on the outside of the membrane, then the rate, the speed at which this molecule is coming in will also increase, okay? Suppose that the concentration of oxygen here was, I don't know, 20, the maximum that could be, okay? And suppose that the concentration of oxygen here was only 5, okay? But the inside concentration was 3 for both sides. Which one would have the fastest rate of transport? If you understand this graph, you would say this guy would have the fastest rate of transport. And why is that? Because the higher the concentration is, look at it, the more higher it correlates with the rate of transport, okay? But that's the case of simple diffusion. You know, life is not so simple, all right? Maybe some molecules get to be lucky enough to have this simple transport mechanism, but other molecules, like glucose, for example, don't, because they can't readily pass through the membrane, and they need help. They need a carrier molecule. And we said this carrier molecule exhibits saturation. How can I show that on the graph? Well, watch this. Glucose also likes to move from an area of high concentration to an area of low concentration, okay? It just requires a carrier to help it move from either side. That's it. Now, the thing is, the rate of transport of glucose in this scenario, or any molecule that uses a carrier really, depends on the saturation of it. Suppose that we're transporting a molecule here to here. We're good. Transporting it here to here. We're good. But then as soon as I hit a specific concentration of that molecule outside of the cell, at that point, I start to saturate my transporter. And when my saturating gets transported, guess what? No matter how much I increase the concentration of that molecule outside of the cell, I will forever be stuck at this saturation limit. You know what this limit is called? This is called the TM, or the transport maximum, which is basically saying that no matter how much I increase my concentration of, for example, glucose outside of the cell, I cannot increase the rate of its transport into the cell past my transport maximum, okay? Because at that point, my transporter is saturated, and we're just going to have to wait for it to be not busy anymore. Alrighty, so that is the kind of example of transport maximum and simple diffusion versus facilitated diffusion, and how you can read this graph, okay? Alrighty, now if you're curious to learn more about this, you can go and watch my last renal video where I actually cover a real-world example of this regarding diabetes. But okay, so these are the majority of the things that I wanted to mention about the actual membrane. There are other stuff that reside here. For example, these proteins over here, they're called CAMs, or cell adhesion molecules. And these cell adhesion molecules, they exactly do what they say they do. They take our cell over here, okay, and make it stick to or adhere to another nearby cell, okay? And you know, we'll talk about these when we talk about the immune system, the immune system actually uses these quite a bit. For example, when some immune systems cells are trying to squeeze out of a blood vessel, they will use a lot of cams, such as ICAMs or VCAMs. Yeah. Okay, the other things I want to talk about is that sometimes on these phospholipids or any of the lipid molecules that are present inside of the membrane, there will be just a sugar residue sitting on top of it, okay? And that's called a glyco, for sugar, lipid. Okay, glycolipid. And other times, some of the proteins that are sitting on the actual membrane will have a sugar residue on them. And at that point, it's called a glycoprotein. Okay? Now, some people would wonder what glycoproteins and glycolipids do. They are part of a bigger facet of the membrane called a glycocalyx. Okay? And the glycocalyx basically includes these glycolipids and glycoproteins, and these guys, they give the cell a kind of a unique membrane identity, okay, as the distribution and presence of certain glycolipids and glycoproteins is different between each cell, okay, and so it kind of gives the cell an identity, and it's kind of like a passport, okay. Sometimes immune systems will actually come and make sure that the cell is indeed our cell and it's not like kind of hijacked or it's like a cancer cell, right? By checking certain glycoproteins or glycolipids on the surface of the cell, okay? They also have other functions, okay? For example, they help with, it actually just keeps the cell hydrated by kind of trapping a layer of kind of water and solutes. And this water layer kind of also can act as a selective barrier, okay? But again, that's a different conversation for a different day, the glycocalyx. But for now, just kind of understand this, okay? But off of this entire discussion, you should have understood that the phospholipid bilayer membrane is selectively permeable, okay? And it is so because of the actual composition of it, okay? And so you can see that if it weren't for the membrane, the cell would have a lot of random things in it, okay? So really, the membrane controls the composition of the inside of the cell, all right? Now that we know all of this, we have to understand the collective influence of certain factors that affect the rate of diffusion of a substance across a membrane, okay? And that is all going to be explained by Fick's laws of diffusion, okay? And these guys they're so simple that I'm just going to kind of breeze through these stuff, okay? Alrighty, so Fick's laws of diffusion, like I said, they talk about the factors that influence the rate of diffusion of any substance across a membrane, okay? And the first one is the concentration gradient. Okay, and we've already touched on this. We've already touched on this multiple times. So let's give it a real world example of this. Okay, and let me change my color here. There we go. So first things first is that molecules like to move down their concentration gradient. So normally the ion sodium, okay, and you guys know there's two types of ions, right? The ones that are positively charged are called cations. Any that is negatively charged, for example, like chloride, is called an anion, okay? And you guys also should know that charged molecules can't just penetrate the membrane, okay? So negatively charged particles or positively charged particles, they can't just penetrate the membrane, so they need specific ion channels, okay? All right, so that was just that little tidbit there. Now, sodium is in a high concentration outside of the cell, and it's in a low concentration inside of the cell. And we'll talk about how this concentration gradient is actually established later, but just trust me for now. And it likes to go from an area of high to low. How does it do this? Well, it uses this transporter, of course, not transporter, the ion channel, but you can see that it's clearly following a concentration gradient. Now, the concentration gradient, all it is a driving force of movement, okay? Because you know molecules are in random motion at all times, continuous random motion. And if you allow them to, they tend to become evenly distributed over time. So for example, if I let the sodium move down its concentration gradient, okay, for a selective amount of time, eventually the concentration of sodium that was outside of the cell will become equal to the concentration of sodium inside of the cell. And at that point, we would have reached a state called steady state or equilibrium or dynamic equilibrium. Okay? What does this word mean? This means that there is no difference in concentration between the sodium inside or outside of the cell, but there actually still is movement going on. A lot of people get this confused. We say that there is no net movement, meaning that the concentration of stuff outside and inside doesn't change, but there still is movement going on. Okay, you just can't see it because the concentrations remain the same. Okay, so that's what happens if I were to let a specific substance move down its concentration gradient for a given period of time. It would reach equilibrium. Let's use another example. Potassium, which is pretty highly concentrated inside of the cell, likes to move out of the cell if we allow it to. If we open a channel for it, it will move outside of the cell down its concentration gradient where it is actually low in. Okay, and again, if we continue to let this happen, this guy will reach equilibrium. All right, and remember, we just said that the concentration gradient is the driving force of movement for these molecules. Okay, they would not be able to move against their concentration gradient. For example, sodium cannot go from an area of high to an area of from area of low concentration to high concentration passively. That would require the use of energy, the use of ATP. Okay, but it can move down its concentration gradient from high to low passively with no ATP or energy required. Okay, so that's one of Fick's laws of diffusion. And this law, all of these laws that I'm going to say, unless I otherwise specify, they apply to every single molecule. Every single molecule. Okay? All right. So that's the law of concentration gradient. The other law is a bit trickier. It's called the electrochemical gradient. Now, I want you to understand this right now. The chemical portion of the electrochemical gradient refers exactly to what we just talked about, the concentration gradient. Okay? This electrochemical gradient, guys, it only ever applies to ions. Only ever applies to ions. So let's take this example. Let's take this example. We'd say that sodium is high outside of the cell, correct? Sodium is high outside of the cell. So let's say there's 1 sodium here, 2 sodium here, 3 sodium here, okay? But the inside of the cell is low in sodium, okay? What does this mean? This means that the outside of the cell, okay, let's say out, in, okay, the outside of the cell is more positive relative to the inside of the cell. We're not saying the cell is negative, okay, although if you want to get technical, it is negative. If I were to actually put a voltage inside of the cell and measure it, it is negative. But in this example, we're saying that the cell is more negative than the outside is, okay, or less positive than the outside. What does this mean? You guys know from like basic high school chemistry that negative charges attract positive charges and vice versa, whereas positive and positive repel each other. So tell me something. If the outside is very positive, okay, and the inside is negative, isn't there an electrical gradient here? Yes, there is. The electrical gradient of sodium is telling it to move inside, go from a very positive area to a more negative area because it's being attracted to there electrically, all right? But there's also a concentration gradient of sodium telling it to move down as well. Okay, so that's two forces that's driving the movement of sodium here. It's electrical gradient and it's chemical or concentration gradient, thus called the electrochemical gradient. Now, notice this. The electrical and the chemical gradient, okay, they don't always have to be working together. All right, they can be working in the same direction or they can be working in the opposite direction. And depending on the delta or the change between the electron and the chemical gradients, you can actually see which one will end up winning. Okay? All right, so that's important to understand the concept of the electrical gradient, and like I said, this only applies to charged particles, mainly ions. Okay, but the concentration gradient applies to everything. Now, what about surface area? This one's very, like, elementary, if you will. If I have a given amount of membrane, I can actually, if I fold that membrane, right? Look at it, if I convolute that same given amount of membrane, right? I can actually fit more transporters on it than if the membrane was just straight. So over here, I can only fit 3 transporters, and I can only transport 3 molecules. Over here, I can fit 5 transporters. I can do 1, 2, 3, 4, and 5. Okay, so this is saying that the higher the surface area, the higher the rate of transport. Same thing with the concentration gradient. The greater the concentration gradient, the greater the rate of transport. Remember, Fick's laws of diffusion are talking about factors that influence the rate of diffusion. Okay? Next thing is thickness or distance, and obviously this should make a lot of sense to you, okay? Let's say I have 2 molecules, exact same size and everything else constant, okay? This molecule has this barrier in front of it, okay? So the distance that it has to travel to get inside of the cell is much further as compared to this guy, who only has to travel this much of a distance as opposed to this much. So the distance that you travel, if it's longer, then the rate of diffusion will be slower. If it's shorter, the rate of diffusion will be faster. Okay, and there are examples of this. For example, certain diseases like, cystic fibrosis, or like, chronic mucus buildup inside of the lungs, for example, can affect the movement of oxygen and CO2 down their concentration gradients across the alveoli, okay? Even interstitial lung disease, for example. But there are real-life examples of this. Okay, the next thing is the solubility, specifically the lipid solubility. And I say this because, remember, the membrane, for the majority of part of it, okay, which is these tails, is hydrophobic, right? And these hydrophobics are just fatty acids, aka lipids. So, you ever heard of the term like dissolves like? Well, yeah, that's the exact same. So if you're lipid soluble, such as O2, such as CO2, you have an easier time moving through this lipid bilayer. And something that is lipid soluble is a lot of steroid hormones, okay, which are made from cholesterol, which is a lipid. And an example of this is like testosterone, or like estrogen, or cortisol, or aldosterone, whatever, okay? All of these guys, they're easily lipid soluble. They can just go through the cell membrane. Something that is not lipid soluble, or very poorly lipid soluble, is glucose. Okay, it requires a carrier in order to actually be able to get across. Okay, great. And that's because, it's water soluble. And then the final thing is the molecular weight of a substance, right? So, and let me finish up here. If a substance is very lipid soluble, then the rate of transfer is obviously faster than a molecule that is not lipid soluble, because guess what? This guy has to go and bind to a carrier. The carrier has to then change its shape and then move this guy over, whereas this dude can just swoop, just fly through the membrane. Now, the molecular weight of a substance, this guy, let's say it weighs less. This guy, let's say it weighs more. Which one do you think is going to have the easier time getting in? Well, the guy that's smaller obviously will easily go through, whereas the guy that's larger takes a little bit longer for it to get through. Okay, so the smaller the molecular weight of a substance, the faster it can diffuse. All right, all of these are Fick's laws of diffusion, okay, and they all apply to every single molecule except this one. This electrochemical gradient, specifically the electrical part, only applies to ions, but the rest of these apply to every single thing. So anytime you're thinking about the diffusion of any sort of molecule, you have to keep these in mind. Okay, great. Now, with this said, we're going to talk about the different kinds of membrane transport that actually exist within a cell. Okay, and you can really categorize it into 2 different things. Okay, one, and let's change color here, being unassisted. Okay, so unassisted membrane transport. And I'll just write it like this. So unassisted versus assisted. Okay? So you can have unassisted membrane transport and assisted membrane transport. Let's talk about unassisted first. In unassisted membrane transport, you can split it up into three. One being the passive diffusion of particles, okay? Of particles, and that's things like, for example, like O2 and CO2. The other one is called the passive diffusion of ions. Now, I know a lot of you are saying, okay, well, ions, you said they need a channel, and that is absolutely very true, but you got to remember that the membrane is actually not assisting that, okay? That's just like a water-filled kind of channel that is just allowing for ions like sodium to just pass through, okay? Whereas a carrier actually has to get involved, change its shape, and all that stuff, okay? All right, so example is like sodium. The last kind of unassisted membrane transport is osmosis, and we'll talk about this in more detail in the next slide, but osmosis refers to the net diffusion of water, okay? So net water diffusion. And guys, I have to mention something to you, that all of these three require no ATP, okay? So with these guys, no ATP is needed. With the assisted membrane transport, there is two types. There is one that's called carrier-mediated transport, okay? And then there's another type. This other one is called vesicular transport, okay? Now, what the hell is carrier-mediated transport? Carrier-mediated has two types. There is facilitated transport or facilitated diffusion, okay? And we talked about this at the start of the video. And there is active transport, okay? Active transport. Now, you can split up active transport into even two different mechanisms, okay? One called primary active, and the other one called secondary active. And I apologize for this, but you can actually split secondary into two types as well, where it can the secondary can actually have antiport and symport. I'll promise I'll explain this. Now, vesicular transport, you can split this into two types as well. One being called exocytosis, the other one called endocytosis. And you can split endocytosis into three parts. One is called receptor mediated endocytosis. The other one is called Pinocytosis. And then the last one, which I don't really have space for, so I'll write it here, is called Phagocytosis. Okay, let's talk about what this side actually means, the assisted membrane transport. So as the name suggests, the membrane has to assist in the transport of some sort of substance here. How does it do this? Okay, with the carrier-mediated transport, as it says, a carrier is involved. And we saw at the beginning of the video what a carrier looks like. It looks like this. It can change its shape. Whereas an ion channel is just this water-filled pore. Okay. Now, carrier-mediated transport can have facilitated diffusion. And make no mistakes, facilitated diffusion has no use of ATP. It is the exact example we've been talking about the whole time, where we're going to take glucose from, for example, the outside of the cell, right, with this carrier, and then it'll bind to its carrier and then open up on the outside and release the glucose into the cell. We'll go through an example in the next slide. The carrier, so the active transport that requires a carrier-mediated transport has two portions, primary and secondary. Primary uses ATP directly, whereas secondary, it uses indirect use of ATP. Okay, so it still needs energy, that's why it's called active, but it's an indirect use of ATP. And it has two methods, is antiport and symport. Symport means a co-transport, meaning I take two molecules and I transport them in the same direction. Antiport or counter-transport means that I'm transporting them in two different directions. Okay, so for example, one is coming inside of the cell while the other one's going outside of the cell. So the same carrier. Okay, so that's that. Now, with the vesicular transport, this is also completely ATP dependent, the whole thing. Okay, and you guys should already kind of be connecting the dots here, because if you guys remember from the very first video, we said that vesicles are carried on these microtubules by specific motor proteins called dynein and kinesin. Okay, and we said for kinesin and dynein to actually bind to and walk across those microtubules, that requires ATP. And not only that, the assembly of a microtubule requires ATP. Okay, so and even the making of these vesicles requires ATP. So a lot of ATP is required in vesicular transport. Okay, and it has two types. One is called exocytosis. The one is called endo. Endo means inside or within. Cyto is the cell. Okay, so inside of the cell. This is a form of vesicular transport where I'm actually bringing a vesicle into the cell. Okay, and there's three types. There is receptor, mediated, where some sort of ligand will bind to its receptor, and then that will kind of pinch off inside of the cell. There's another one called Pinocytosis, where it is non-selective, and the cell is pretty much just drinking the interstitial fluid surrounding it and taking up any random molecule. And then the other one is called Phagocytosis, which is actually very extremely selective and specific. Okay, and this translates directly to cell eating. Okay, phago means eating. Okay, now exocytosis is the exact opposite of endocytosis, where exo meaning outside and cyto again means cell. So outside of the cell, this is where we are taking a vesicle and pushing it outside of the cell. Okay, and now we're going to talk about each of these in depth and give an example of all of them. But before we do that, we have to talk for a second about these bad boys. Cell-to-cell junctions, specifically specialized cell-to-cell junctions. Okay, what are they and what do they do? We have three main types of cell-to-cell junctions, and I've put them all in the same picture here for educational purposes, but do remember that they don't necessarily all have to be on the same cell. Okay, so the first one is going to be this guy over here. Okay, it's a tight, and let's zoom in actually and rewrite this because I want it to be neat. So it's called a tight junction. Okay, and tight junctions, guys, they are pretty much holding adjacent cells very firmly together. Okay, and they're forming an impermeable barrier almost. Okay, where would you find this tight junction a lot? You'd find it very much so in the they call it enterocytes of the small intestine. Okay, and the small intestine, it's where the site where most of the food absorption occurs. Let's say there's some sort of toxin in your food. Okay, and it's just this toxin is kind of just floating around, and normally we have specific protein channels and these membranes to kind of prevent them from going into the blood on this other side, right? But what if they could just kind of squeeze in between these two adjacent cells, and then they make their way into the blood? That's not good, right? How do we prevent that? Well, we prevent that by the presence of these tight junctions, okay? They are sealing off the outside of the cell, okay? So that things don't just kind of move down here into this basolateral membrane. Basolateral means just the portion of the cell's membrane that is facing the blood, and go into the blood and cause danger to our body, okay? They also function in preventing the cells from migrating, okay? All right. The next kind of cell-to-cell adhesion or junction is called a desmosome, okay? And it's this guy right here, okay? So this whole thing from here to here, it's called a desmosome. And you'll see that a desmosome consists of many parts, okay? One is the plaque, okay? The other one is these proteins over here called cadherins, okay? Cadherins. And there is this one other portion, which are intermediate filaments. And you guys, if you remember, we said the intermediate filaments inside of the cell are kind of there to help resist mechanical tension or stress. And a good example of this is keratin. Okay. Now, these desmosomes, they are very, very abundant in tissues that are like subject to considerable stretching. So they're very much so abundant in skin cells, in the heart, right? For, the heart has to pretty much beat like a gajillion times every single day for the rest of your life. So, and that's going to stretch the heart continuously. And so, yeah, the skin as well, right? And a woman's uterus during birth, right? It has to expand a lot and stretch a lot. So it definitely requires these desmosomes. They are the strongest cell-to-cell connection by far, okay? That's why they have to be present in cells that are subject to a lot of stretching, okay? The final type of cell-to-cell junction is actually called a gap junction, okay? So here it is. It's called a gap junction. These ones are very interesting. They're very interesting because essentially you can think of them as communicating junctions where they permit small kind of water-soluble molecules or particles like ions to pass between these two connected cells. So here's the cytosol of one cell and here's the cytosol of another cell, right? And they're going to just allow for this positive charge to move. So for example, let's say you have sodium inside of the cell, right? That is kind of activating the cell and leading to contraction. We'll talk about it in muscle physiology, but this positive charge can flow from this cell to this other cell and make this cell positive to the point where it can cause it to also contract if these two were, for example, like muscle cells. These gap junctions, due to this function of passing ions and communicating a electrical charge that might help contractile activity, are very much so present in cardiac cells, so cardiac muscle, and smooth muscle cells, but very special smooth muscle cells, okay? But essentially they allow for the movement of ions to transmit electrical activity, which allows for kind of a synchronized contraction, okay? Now, these gap junctions, they're made up of these two proteins called connexons. And fun fact, one connexon is made up of 6 connexins, okay? Alrighty, but again, they just form this kind of small water-filled pore that allows for small ions to pass through. That's it, okay? So these are your junctions. Okay, now that we talked about all this, we can go ahead and talk about all of our actual forms of membrane transport, okay? So first things first, we talk about oxygen, okay? We know that it'll move from area of high concentration to an area of low concentration, and we know that CO2 is in high concentration inside of the cell, so it'll move outside, okay? CO2, okay, great. These two, both lipid soluble, and they will move past the membrane very easily. This, as we classified in the last slide, is called passive diffusion of particles. Okay, passive diffusion of particles. Easy peasy. And again, remember the concept of equilibrium, right? So that if we allow this to keep going on, they will equilibrate. Okay, this oxygen would equilibrate with the outside, and they would reach no net diffusion, meaning that still things are still moving, but it looks like they're not moving because the concentrations on either side of the membrane are the same. Okay. What about the other part where we said there are passive diffusions of ions? Okay, so this one, passive ion diffusion. And this is absolutely true. We may have specific ions such as sodium, right, which are in high concentration outside, but are in low concentration inside. Okay, and we know that the inside of the cell is usually more negative than the outside, so there is an electrochemical gradient pushing this ion inside of the cell through its specific channel. Okay, this is a sodium specific channel. Okay, okay. And again, once again, why does it need a channel? It's because positive or negatively charged substances cannot cross the plasma membrane, okay? All right. The other thing we talked about was facilitated diffusion. Let's get rid of this. So let's write it down here. We said there is facilitated diffusion. And what does that look like? Like I said, let's say we have this molecule of glucose, all right? So high glucose outside, low glucose outside. How does this glucose molecule get in? Well, guess what? It can't readily permeate the membrane because it's water soluble. It needs a carrier protein. So here's what a carrier protein will do. Watch. It will allow for this guy to actually come in and bind to the binding site. Once this guy binds, it'll cause this protein, the carrier protein, to change its shape. All right, so now it looks like this, and the glucose molecule is inside of it still. Okay, and eventually, this glucose molecule, once this guy changes its shape fully and opens up on the other side, will dissociate. Okay, and now this glucose molecule has finally moved into the cell from high to low, and after this guy unbinds, this guy goes back to its original shape, facing the outside again. Okay, beautiful. That's how we move glucose in. That's called facilitated diffusion, okay, or facilitated transport. Awesome, awesome. Now, the next thing that I want to talk about is osmosis, and I really want to talk about this in quite a bit of detail here, so bear with me. Okay, now with osmosis, it's just talking about the net diffusion of water, but water can move in two ways. Okay, let's say that the outside has a high concentration of water and the inside has a low concentration of water. Okay, this means that water can readily move from high to low, but can water move through the plasma membrane? And the question that a lot of people face is that, okay, well, the inside is hydrophobic, right? So water hating. So probably no. But it's not truly correct. Large water-soluble molecules or charged molecules really have a hard time going through this area, but water itself, it is a small particle, so it can sometimes slip through these actual phospholipid molecules and get in there. Okay, but this is not very common for it to occur. Okay, but the force that is actually moving the water is its concentration gradient. Okay, high to low. But the water can actually get in from a more controlled manner, and that is through these protein channels called aqua porins. And again, it's in the name. Aqua means water. Porins is just like this hole in the membrane. Okay, and you can see water goes from a high concentration to a low concentration. Easy peasy. That's the first driving force for water. But you know something else? There's also another driving force for water. There are solutes, okay, that can't readily penetrate the membrane. Which means what? Which means that these molecules, right, these salts, if you will, they have a hard time going through the membrane themselves, right? They're going to try to go through the membrane, but they get repelled, all right? We call these guys non-penetrating solutes because they can't penetrate the membrane. They need some sort of, I don't know, ion channel or carrier or whatever, okay? Water has this special property where it likes to move from an area of low solute concentration, specifically low non-penetrating solute concentration, to an area of high penetrating solute concentration. So watch this. 1, 2, 3, 4 non-penetrating solutes here. But there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 non-penetrating solutes. Notice how the water is moving from an area of high water concentration and low solute concentration to an area of low water concentration and high solute concentration, okay? The water always loves to do this. It's just a property of water, okay? Now, the reason why this is happening is because these non-penetrating solutes exert an osmotic pressure, okay? This osmotic pressure is a pulling pressure, which kind of pulls water towards them, okay? The best way to think of it is like this. Because these non-penetrating solutes can't actually equilibrate, okay, and go and meet the water on the other side, and why can't they equilibrate? Is because they can't penetrate the membrane. So you know what they do in spite? They actually pull the water towards them. They say, hey, if I can't go and be with the water's coming to me. So there's really two forces that actually drive movement of water. First is the concentration gradient. Second is the actual osmotic pressure of these non-penetrating solids. This, my friends, is called osmosis. Okay, so that's osmosis covered. All right, next is going to be primary active transport. All right, so let's talk about primary active transport. Okay, now this one's going to be a little tough, so bear with me here, but it's nothing we can't go through. Primary active transport. Okay, so this is the original conformation of this carrier, okay? And by the way, one of the main examples of primary active transport in the body is shown by a classic enzyme carrier molecule called sodium/potassium ATP pump, or ATPase. We'll talk about why it's a pump, and we'll talk about why it's an ATPase, okay? Active transport is defined by what? Active transport is defined by taking a molecule and pushing it uphill against its concentration gradient. So let's take an example that we've been using so far. We know that sodium is usually in a high concentration outside of the cell, and it can readily, in the presence of a channel, come down its concentration gradient into the cell. But what if I wanted to take the sodium from a low concentration and push it out to somewhere that has a high concentration gradient? Well, this would be going against the original concentration gradient. Well, how would I do this? I need energy. I need ATP. This is where primary active transport comes in, by pushing things against their gradient. So in this current stage of configuration, there are 3 high affinity binding sites for sodium. Okay, so I have 1 sodium, 2 sodium, 3 sodium. And guess what? They will all come and bind to these binding sites. Okay, actually, you know what? Let's draw it neatly. So boom and boom. These 3 are high affinity binding sites for sodium in this configuration of the protein. Okay, as soon as the sodium binds, you know what ends up happening? This molecule, this sodium-potassium ATPase pump, will do something pretty crazy, where it will take a molecule of ATP, adenosine triphosphate, and it will break it down into an adenosine diphosphate plus a phosphate molecule, which actually ends up being bound to this carrier molecule now. Okay, so the phosphate becomes bound to the carrier molecule, and that's shown by this. Okay, so let's take a quick recap. In this configuration, there are 3 high affinity sodium binding sites, where sodium will go and bind to these binding sites, all right, and the ATPase function will then kick in, where the sodium-potassium pump, ATPase, will break down ATP into ADP and a phosphate, and the phosphate group will then attach to the actual carrier. Okay, the phosphorylation, okay, and you know, we can show like this. Here's our sodium molecules just chilling in here now. Okay, the phosphorylation makes it so that this protein carrier molecule, okay, ends up changing its shape, and it flips from this configuration into this configuration, all because of this added phosphate molecule. Okay, and now in this configuration where we have changed the shape of this protein, those high affinity binding sites that were available for sodium, they're no longer high affinity. And so what ends up happening is that these guys will now leave, okay? And we have now successfully pushed sodium from an area of low concentration to an area of high sodium concentration, okay? So here sodium comes out here. Great. Now, something else is going to come in though, because in this configuration, there are two high affinity binding sites for potassium. So watch this. Two will come and bind. And this is the funny part. As soon as these two potassium ions come and bind to this high affinity binding site, what ends up happening is this phosphate group that was attached here from the ATP will immediately leave, okay? That phosphate will leave, and if the phosphate leaves, guess what happens? This molecule becomes dephosphorylated and flips back to its original shape. And when it flips back to its original shape, look at what happens. It opens up like this, and those two high affinity binding sites are no longer high affinity in this configuration, and they let go of the potassium, okay? And they bring potassium here, all right? Now, what happened with the potassium? Remember, potassium is usually high inside of the cell. So we took potassium from a low concentration to a high concentration. Again, we pushed both of these guys against their concentration gradient. Now, this is where that cycle ends, right? So at this point, we released the potassium, but now we have these binding sites open again for high affinity sodiums to come in and bind, and then the cycle repeats. But here's the key point. Remember how way back in this slide, I said that there's a reason why the concentration gradient exists this way, why there's high sodium outside and high potassium inside? The reason is this sodium-potassium ATPase pump. These guys establish this gradient by pushing sodium from inside of the cell outside. They eventually create a gradient where sodium is super high outside and super low inside, okay? And the same thing with potassium. They keep taking the potassium from outside of the cell and pushing it in the cell to make this guy super high inside of the cell, but super low outside of the cell. Okay? Great. And another thing, remember how I said the inside of the cell is negative usually? It has a net negative charge. It's because of these guys as well. All right? If you notice, I push out 3 positive charges, but then I bring in 2 positive charges only. So I have a net negative movement, which means that over time, when these guys keep working and working and working, my cell will start to become more negative, okay, in charge. Okay, and so that's primary active transport with the sodium-potassium ATPases. And these bad boys are placed in every single cell in your body, okay? Now let's talk about secondary active transport. Secondary active, and guys, the reason why it's called secondary active is because it is using the gradient established by these sodium-potassium ATPases to move things in and out of the cell. That's why it's called an indirect use of ATP, because the ATP was actually used here to establish the gradient for the sodium ion, and then that is used to transport stuff. So let me give you an example that makes sense to you, okay? So the first one is going to be called a symporter, which we said means to transport things in the same direction, and the other one is an antiporter, which means things go the opposite direction. A very common example of this is in the GI tract, where in the lumen, okay, where, and you know, let's draw it like this, where we want to absorb glucose, okay? But glucose is in low concentration in the lumen, but it's normally in the high concentration inside of the cell, okay? So how do we absorb it? We obviously need energy to move something from low to high, right? Well, guess what? Sodium is usually in high concentration outside of the cell, and it'll be super kind and go down in from this channel into the cell down a concentration gradient, and then glucose will see this and he's like, hey buddy, you just moved down your concentration gradient, can I just hop off the back of you and come with you? And he will do exactly just that, and that's how we move the sodium in inside of the GI tract, and even in the kidneys when we want to reabsorb glucose, okay? So that's how glucose moves in the GI tract and in the kidneys, okay? And this specific transporter is called a sodium and glucose co-transporter, okay? So sodium glucose co-transporter, or symporter, all right? And it's a form of secondary active transport. The other way is let's say again, sodium is moving down its concentration gradient inside of the cell. Okay, nothing crazy. But we want to, oh, this is a good example. We want to move another famous ion called calcium. But instead of moving it in the cell, we want to move it outside of the cell. You know, usually calcium is in a low concentration inside of the cell, and it's pretty high outside. So calcium sees sodium moving in. It says, hey, you know what? I'm going to use you moving in for me to move out, okay? And that in that scenario, that's called an antiporter, okay, where it's moving against the concentration gradient. But it is using the energy of sodium moving down, okay? All right, so that's secondary active transport. Alrighty, we've pretty much talked about almost everything here. The last thing I want to talk about is exocytosis and endocytosis, okay? Let's talk about exocytosis first because it's much easier and much more brief, okay? So with exocytosis, if you guys remember, right, we were talking about vesicles. So I have to take you back to cell biology a little bit. Remember that, you know, from the nucleus, we synthesized an RNA, and then the RNA goes to the ribosomes on the ER, which make a protein. The protein will get kind of folded a little bit, about packaged in the ER. The ER will send the vesicle into the Golgi. Then the Golgi will modify it, package it, add some signals on it, okay, and then send it out. Where is it sending it out to? Here's what we can zoom in on, okay? The Golgi will package this molecule, okay, into a giant vesicle, and let's say this molecule is actually a hormone. Let's say it's some sort of hormone. Let's say it's insulin. Okay, boom, boom, boom. Let's say this guy is insulin. And on the surfaces of this actual vesicle, there will be specific docking markers or targeting sequences that tell it, hey, you are destined to be, for example, excreted outside of the cell, okay? Or you're destined to be put on the cell membrane, okay? That's what exocytosis is really used for. It's either used for secreting something outside of the cell, like a hormone, or incorporating something into the membrane, like a transporter, an enzyme, or a carrier, or whatever. So, remember that we said this guy's a docking marker acceptor and this guy's a docking marker? Well, now we're going to give them more specific names. We're going to say that this guy that is on the actual surface of the vesicle is called a V snare, okay? And this guy, the docking marker acceptor on the membrane, is called a T snare. Now, before you freak out, remember, V stands for vesicle. It's on the surface of the vesicle. T stands for target. It is on our target surface, okay? The target being the cell membrane. And I told you that when these two kind of come together, they kind of click onto one another, and it allows for this membrane of the vesicle to fuse with the membrane of the cell. This is what it will look like. Here you can see it in action, where the membrane of the vesicle is literally fusing with the membrane of the cell, and it's opening up the inside bubble, okay? Where now we can actually release our insulin molecule, our hormone, okay? Boom, hormone's going out. And let's say there was, like, you know, a bloodstream here. That insulin will eventually find its way into that bloodstream. Okay? Beautiful. This is the process of exocytosis. And I said it's used if we want to secrete molecules such as proteins, okay, into the cell, or sorry, outside of the cell, or if we want to incorporate something into the membrane. All right. That's the process of exocytosis. Exo meaning exit. Okay, pushing something out. Now let's talk about endocytosis, okay? And we know there's 3 types of endocytosis. The first one is called Pinocytosis, okay? And this is done by specific cells, okay? A specific cell, an immune cell called a dendritic cell, actually goes through the process of macropinocytosis. So pino means to drink, cytosis means cell, right? So cell drinking, macro meaning large. The dendritic cells go through this process of pinocytosis, but specifically they go through macropinocytosis, where it's called large cellular drinking. And they're just kind of taking a non-selective, and this is key here, they're taking a non-selective sample of anything that is inside of the interstitial fluid. Okay, so look at it. The membrane kind of invaginates inwards, okay, and it's taking kind of any of the fluid and particles that were inside of that interstitial fluid and non-selectively just picks it up, okay, and then the membrane kind of pinches off, right, and when the membrane pinches off, it turns into this little vesicle, okay, that was containing all of our little things, okay. And as soon as this dude comes in, it comes and interacts with the garbage can of the cell, which is our what? Our lysosome. This vesicle will immediately become in contact with the lysosome. And you guys remember the lysosome has hydrolytic enzymes that will do what? It will degrade any of the substances that are inside of this actual endocytic vesicle is what they call it, okay? All right, that's that. Now, that was macropinocytosis or pinocytosis, non-selective, and an example of a cell that does this is dendritic cells. We'll talk about them in a bit more detail. The other kind of endocytosis is called phagocytosis. Phago meaning eating. Okay, so cell eating, and there are specialized cells that do this, and I mean only specialized cells. There's two immune cells that do it. They're called macrophages and neutrophils. Macrophage, it's literally in the name. Macro meaning large, phage meaning eater, large eater. That's exactly what this cell does, just eats. And it literally does this. It takes these, the plasma membrane, right, and it forms like this kind of arm shape. And after it forms this arm, it kind of encloses its arms together eventually and captures like a microbe or a bacteria, okay? And after it captures this microbe or bacteria, right, it doesn't even have to be bacteria. Sometimes it's just like a protein, but more often than not in an immune system case, it's a microbe. It captures this guy and eats it, or literally brings it in, okay? And that's called phagocytosis. Now, this process is extremely selective because only like 2 cells can do it, okay? And these arms that this guy forms are called pseudopods, okay? It forms these arms around whatever it's about to eat, whether it's a microbe or it can even be worn out red blood cells. So you guys know in the spleen, we actually remove dead red blood cells. That's done by macrophages. They literally phagocytose old red blood cells and they just kind of eat them and degrade them, okay? And the exact same thing happens. Watch. Boom. Exact same thing happens. Take this guy and we, oops. Okay, so I redrew this. So yeah, with the phagocytosis, right, we take this kind of a thing in, right? It pinches off into a vesicle immediately, and then that vesicle will then go ahead and fuse with the lysosome, which will just destroy any bacteria or red blood cell or protein or whatever, any foreign substance that we're taking in. It will immediately be destroyed, okay? Alrighty. The last one is this process of receptor mediated endocytosis, okay? And so imagine this membrane, right? We have just like normal receptors. And the ligand will come and bind to its receptor, and as soon as it binds to its receptor, you know what happens? These molecules called, in blue, right? In blue, called clathrins, will immediately start coating the surface of the cell membrane and forming a pit. This pit will then be called a clathrin-coated pit. Okay, clathrin-coated pit. And these guys will start to kind of pull on this membrane, all right, until it pinches off with the receptors and the ligands in there, okay? And once this guy pinches off, you're not going to believe it. It will then form a endocytic vesicle with those receptors still in there, okay? Still having their ligands and everything, and still coated with that clathrin coated pit, okay? Boom. And you know there's other steps involved here, but eventually this same dude here will interact with a lysosome, okay, and the lysosome will come and break down any materials that reside inside of this actual endosome, okay. Boom. So that's 3 processes of endocytosis. There is macropinocytosis, there's phagocytosis, and there is receptor mediated endocytosis. And this is pretty much occurring with anything that binds to a receptor. So insulin, iron, vitamin B12, okay. Another good example of this is like the COVID virus, right? Where you guys know the spike proteins on the actual COVID virus will bind to the ACE2 receptors on the membrane of, for example, the lung, okay? And then it'll kind of trick the cell into using this receptor-mediated endocytosis process to kind of take it in, and then the virus will start replicating inside of the cell that way, okay? So this is just a real-world example of how this works. Okay. Last thing that I want to talk about is how is it that with exocytosis, we add a piece to the membrane, and with all of these endocytosis processes, we take pieces of the membrane away. Shouldn't this kind of like, make the membrane unstable? And it's true, the rate of these processes are highly regulated to maintain a constant membrane surface area. In fact, greater than 100% of the plasma membrane is used up in an hour to wrap internalized vesicles, okay? And so the membrane is constantly restored, retrieved, and recycled. And yeah, that's like a very, very important thing that occurs. And lastly, since we said everything about these guys, we can say that this receptor-mediated endocytosis is also pretty selective because it does depend on the receptor, and we know that receptors are very selective as to their ligand, right? For example, something that is the shape of a square can't bind to this triangular shape. It has to be triangle, okay? And pinocytosis and receptor-mediated endocytosis can be done by other cells. Phagocytosis is done specifically by macrophages and neutrophils, and macropinocytosis is done by dendritic cells. Okay, guys, this was such a long video, and if you stuck around this far and if you understood everything we talked about, thank you so much. Please consider leaving a comment if you've stuck this far, and like and subscribe if you truly learned something, and I'll see you in the next video. Thank you so much.